keeping with the theme of different speaker and different title, I'm not Jay Clark. I'm Anthony. I lead R&D at Arima Genomics, and the title of the talk today is Rapid and Sensitive Discovery of Genomic Interactions Using Arima IC. So I'd like to start with the acknowledgments in the beginning. I wanted to thank the NIH, specifically the NHGRI, for funding for the development of the High-C kit that I'll be telling you a little bit about today. I also want to thank the organizing committee. We've had a lot of really good discussions here. It's been a beautiful venue, and it was pretty enlightening seeing ancient Olympia today. So I'm going to break this talk out into a few different parts. Super brief introduction. I'm going to dive a little bit into the Arima High-C protocol and some of the nuts and bolts there. Some of the analytical benchmarking that we've done regarding chromatin looping analyses. I want to spend some time talking about our optimized low input High-C protocols and then some future directions. So, super brief, we've had a lot of really exciting talks across all scales of genome organization, from sort of the most zoomed out view all the way to the most zoomed in view in terms of chromatin loops and CTCF mediated loops that we've heard about today. High-C is a ligation-based method to detect these pairwise interactions, and it's not a ligation-free method like GAN that Anna talked about earlier, but it is useful for measuring pairwise contacts genome-wide. So just jumping right into the goals for the Arima High-C kit, we started out really striving to do several things, one of which was to reduce the sample input requirements, to hopefully open up access to low input sample types that were previously challenging to do with High-C. We wanted to simplify the protocol further and also make it faster. And then just from my own personal experience in Bing Ren's lab where I did my Ph.D., it was always a little bit frustrating, sort of the lack of really quantitative and predictive QC, so we wanted to build that in so that you can sort of confirm library quality via simple QC assays, improve signal to noise, and maybe most importantly, try to bring down the cost of the sequencing requirements via a very high quality library. So a little bit about the High-C protocol. I'm not going to go into this in too much detail. It's largely similar to what you've seen already. Some of the key differences in this digestion, fill-in, and ligation process is that we use a cocktail of restriction enzymes that increase the coverage of the genome, increase the cut site density, and the digestion efficiency during the High-C process. And this part of the protocol here, which may take 2 days in other protocols, is whittled down to about 6 hours. So this process here is 8 simple steps. It's a single tube High-C chemistry, which means when you put your cells into the lysis buffer, there's no spinning, there's no tube changes. You just keep adding on reagents, and the chemistry has been optimized to work in the presence of the lysis buffer, which is helpful for low input applications when you really don't want to be transferring tubes or doing extra types of manipulations. Like I already said, multiple restriction enzymes and the QC assays. So what I'm just showing here is after the typical High-C process, and you have your purified ligation products here, there's a QC checkpoint, which I'm not going to go into details on how it works, but essentially the value that you obtain here, simply using a qubit, is predictive of the quality of the final library when you sequence. So after High-C, it's just the standard biotin enrichment, adapter ligation, PCR and sequencing. And the way that we do this is not trying to reinvent the wheel on library prep, just to support a lot of the commercial library prep kits that are on the market that are already quite good. In terms of chromatin looping analyses, when we set out to benchmark the technology, we tried to think of a data set in a sample type that would be accessible and widely used. So we benchmarked all of our data to the in situ high-seq data published in the lab from Reza's lab. What I'm showing here is a snapshot of a 1 megabase locus in GM12878. This is from one of the replicates from that data set that was sequenced to about 3.6 billion raw high-seq reads. This is an ENCODE cell line. So we generated Arima high-seq data using the protocol I just described, sequenced this to about 1.2 billion reads, or a third of the depth, and after processing the data through Juicer, calling loops via hiccups as part of the Juicer framework. One of the things we noticed when we started to visually inspect the matrix was that there were these frequent examples where the loop pixels that would support a particular loop call appear to be more enriched in our data set compared to the published in situ high-seq data set, despite there being half the number of high-seq contacts that were actually going into the map. So one of the things that could result from this is that because the signal is enriched at these loop pixels, maybe you're able to detect loops more sensitively or more loops at a genome-wide scale. And that turned out to be the case. So here's the number of loops that are called in the published in situ high-seq data set. This is the number of loops called from 1 billion reads in our data set, processed using the exact same pipeline and the exact same parameters using Juicer. And then when you downsample that data, even down to 600 million reads, the number of loops that you can identify is over 50% more. Just to show you that this isn't a cherry-picked example from internal data from a customer who generated Arima high-seq data in IMR90 cells compared to the published in situ high-seq data in IMR90. This was sequenced to 700 million raw reads, and the same sort of effect. You do see a decline in the overall number of loop calls. Even down to 400 million reads, you're still identifying about 9,000 with a 70% overlap between the two data sets. So one of the things that we thought is just the total number of loops called isn't necessarily the end-all be-all, but how well are we recapitulating the loops that were called in the in situ high-seq data. And it turns out that in our GM12878 data, that's about 80%, but there's about 9,000 loops total that are unique to the Arima high-seq data. So we're interested in exploring that a little bit more. So when we look at these 9,000 loops, we do an APA plot, which is essentially taking the Arima high-seq unique loop positions and overlaying them on the Arima high-seq map. You would get something that is expected that looks like this, with a strong pixel enrichment at those loop positions in the Arima high-seq map. And if we were identifying loops that were completely devoid of signal in the published in situ high-seq data, we would expect to see something that was more or less blank. But it turns out that on average, the intensity of those loop pixels that are called in the Arima high-seq data, when you look at the published in situ high-seq data, is just a little bit weaker, suggesting that the signal intensity at those loop pixels is enriched in the Arima high-seq data. It brings us over the threshold of detection, and it enables you to identify them. Switching gears to low input high-seq, I want to talk you through an experiment that we did. I think this is similar to what others have done who have published low input high-seq protocols. So we took 1 million cells and then diluted it down to 1,000 cells by half log orders. And this is just plotting the total amount of DNA that you can recover after doing high-seq, just extracting the DNA after ligation. And some of the values here are expected, about 3 micrograms for 1 million cells, and as you go down to about 50,000 cells, you're in the 75 nanogram range, and 1,000 cells is actually below the limit of detection on our qubit. So that's a sense of how much DNA that we're actually working with within those cells. I like to think of complexity quite simply, just in terms of how many PCR cycles are required for amplification. Internally at Arima, if a library takes 8 cycles or less, we think that that's sufficiently complex to sequence deep enough to detect loops from that particular library. In the low input experiment that we had here, if you start with about 50,000 cells, a single library can get you that type of loop level complexity, and about 10,000 cells to give you what we would consider a tag level complexity with about 25 KB bin size. And probably most importantly, this is all great, but when you actually sequence the data and you plot the distribution of long-range cis contacts or short-range cis contacts or interchromosomal contacts, the relative distribution of those types of contacts is almost identical from a million cells down to 1,000 cells. So the general properties of the data are quite conserved across 1,000 fold. We sequenced the 50,000 cell Arima Hi-C libraries deeper, and again we compare it to the in situ Hi-C data. This is just a locus, about 2 megabases in the published data set. We sequenced these to about 800 million reads and 350 million valid Hi-C contacts, and overall the data look quite similar. And when we process the data using Juicer and hiccups to identify those loops, the total number of loop calls are quite similar, with just a little bit more in the 50,000 cell Arima data. Very briefly, I'm just going to talk a little bit about high chip. I think we've heard some very useful discussion about thinking about high chip and the interpretation of the data, and I would say the maturing tools to analyze the data. It's something that we are interested in, as well as captured Hi-C. I'm going to show just some very preliminary data that we've generated using high chip. For those who don't know, I'll just go through this very quickly. It's just like Hi-C from the digestion through ligation, except adding a chip step here to IP your protein factor of interest and also capture that ligation junction once it ends up getting sequenced. So ideally, it will measure the 1D protein localization in the genome via the sort of 1D chip signal, but also the 3D interactions that are mediated by the protein of interest, such as H3K27 acylation. This first snapshot here was from 1 million cells, and the boxes in blue were the loops identified. I think this particular analysis was done quite a while ago. We were using HiCups for that, but currently we're evaluating maps from Yinghu's lab as well as FitHiChip from Ferhade's lab. This is just illustrating this example of really like a very long-range enhancer-promoter interaction. I think this one is about 600 kb that kind of encases the entire locus, and this enhancer here is also involved in other significant contacts with other putative enhancer marks there. So I am going to conclude just by summarizing, I think, some of the key points. I hope that I was able to show you and introduce you to our rapid 6-hour Hi-C protocol and the enriched looping signal that it can produce. And I think one of the most important takeaways is that the enriched looping signal enables a more sensitive loop detection from significantly fewer reads. Our protocol is optimized for low input cells. And just as a general sort of philosophy about us, we really strive to be a simple and flexible Hi-C protocol that enables multiple sort of C assays like Hi-C, HiChip, and capture Hi-C, as well as multiple applications. I was hoping to have a little bit more time to talk about genomic rearrangement analysis. I think I saw some posters out there that were along those lines, and we're also interested in the kind of unique ability Hi-C has at detecting genomic rearrangements in cancer and using the Hi-C data to analyze the impact that those rearrangements have on protein architecture and gene regulation. With that, I'd like to thank those at Arima who did a lot of this work, And with that, I will take any questions. Interesting.
Hey, thanks. That was really fascinating. And I mean, obviously, you guys have been doing a tremendous amount to democratize these technologies, which is really, really good for the field. I did have one question for you, which is, there are facts of the matter about how frequently bits of the genome are in contact with one another, and it does seem to be the case that the differences between the Arima protocol and some of the openly available protocols from my lab and other labs, those differences are salient in terms of saying, well, the probability that these two things are in contact are actually different with one another. My general impression, I haven't seen a lot of data, but my general impression is that intrachromosomal contacts are much more frequent in Arima data, and interchromosomal contacts, which are used for things like compartment polling, rearrangement detection, etc., are less frequent. But again, there are facts of the matter about that, and there are some biological settings where the only thing that you see are changes in the inter to intrachromosomal ratio. So I wanted you to maybe comment about how can users of the Arima kit think about that. How can you be sure that what you're getting accurately reflects what's going on in the nucleus? Can you comment a little bit if there are some sort of generic biases, and maybe all the protocols have them, but if there are ones that are particular to the Arima kit, what are they? How do you think about them? How do you avoid situations where they could confound your biological interpretation?
Yeah. So you said that the Arima data has more intrachromosomal contacts?
General impression, I mean, I haven't made like a study of it.
Okay.
And obviously there's a fixed number of contacts, so they have to go somewhere.
Yeah, I think it's a good question. One of the things that we think is driving the enriched looping signal, but also potentially that, is that we get through the protocol a little bit faster, so there's less overall time exposure to detergents and heat that you might imagine would cause the structure to dilapidate a little bit. That's one speculation.
So I want to push back on that because I don't think that's it.
Okay. Do you have another hypothesis?
I don't know, and actually this is of great interest, right? Because there are general differences between the maps, and one of the things that's tricky, and I mean, again, there's tremendous credit that goes to Arima for putting out a kit that many people have had success with, but there are these significant statistical differences hard to debug, right? If you're an external person who sees this data set, and it looks different than the data sets that you've seen before, it's hard to debug and say, like, hey, I don't quite know why that happened. So that's a challenge, I think, that the community faces.
Sure.
Two very quick questions, I promise. Okay. So the first one is, how flexible is it for difficult cells? So for example, we do a lot of thymocyte work and we have to really play with the conditions because they're such compact chromatin. So.
I guess just speaking from the variety of sample types that I've seen our customers using, it seemed relatively robust to different cell types, but I think if that is something specific that you've had to deal with, it's possible that you might have to optimize.
That is possible. It's not one tube that you can only do this. There are possibilities.
The idea would be that it would be robust enough to work on the first try without optimization.
The second question, super different.
Sure.
Are the restriction enzymes a trade secret? Because for Capture-C, you need to know that information. So
the cut sites that are GATC and GANTC, we can provide the locations, and that's key for all analysis. So that comes up.
Thank you very much.
Sure.